Okay, well, thank you all for joining our webinar today. We are really excited to hear about frogs from Helen Willsey from University of San Francisco. And she's going to be talking to us about her work with CHD2 and the frog model. So Helen, I'll let you take over. Thank you.
Yeah. Hi, I'm Helen. It's wonderful to be here. Thank you for the invitation and for coming or watching. I'm an assistant professor at UCSF and also an investigator with the Chan Zuckerberg Biohub. A little bit about me. I grew up in Tennessee. I went to Duke University to get a bachelor's in biology. I then went to Yale University to get my PhD in genetics. And then I did a postdoctoral fellowship at UC Berkeley and UCSF, where I really developed this frog model to be able to study many different genes in parallel to understand what they do during brain development. And so the work that I'll tell you about today, I've obviously centered it on CHD2, but it's part of a larger effort that my lab is working on now to study genes that have been associated with autism spectrum disorders. So obviously there's a lot of other things that CHD2 does, but that's how I got interested in it. And so at the time that I started this work, CHD2 was one of the top genes associated with autism. And so we studied the top 10 of those genes and found out a lot about them. And so what I'll tell you today is a part of that story about CHD2, and then what we're now planning to do in my lab to really start focusing in on CHD2 more specifically. Okay. Okay, great. So this is, and you'll see in my background and here now an example of one of our frog images. And so the first thing you might say is, well, what can we learn about frogs that will actually be relevant to what we care about, namely the function of CHD2 in humans? And so what I want to point out is that actually, when you look at a variety of different, what we call model organisms, like you might know, a lot of people use mice to study processes that happen in human development, right? What you may not know is that the processes that go on in frog development, or with Xenopus, which is the genus, they're actually very similar. So they go through, fertilization of the egg, a process called gastrulation, looks a lot like humans. Then they go through this stage where actually us as embryos look a lot like a tadpole. And actually a lot of the same processes that go into building a brain of a human are the same processes that go into building the brain of a frog. Now, of course, it's much more complicated in humans and there are important differences. But when we're starting out with a basic question of what does this gene do, where is it expressed, what happens if it's lost, we can actually start to build really foundational knowledge in frogs that we then can go into more complex systems and validate or learn more about. Furthermore, for a gene like CHD2, what's important to remember is that actually when you look at the sequence of CHD2, and of course the DNA then codes for RNA and the RNA codes for the protein. So here I'm showing you a protein sequence for CHD2. If you actually look at the sequence and the code of the protein, the version that's in frog, which is this lower line, is actually very similar to that in the human, which is the upper line, which is just part of the gene. So actually at the protein level, there's almost 80% identical sequences between frogs and humans. So it's been highly conserved over evolutionary time. And so this means that most likely the things that CHD2 is doing in frog cells and in frog development are probably the same things that it's doing in human development, though of course it can be more complicated in the human. But we can build, again, foundational knowledge about what this gene does by studying it in frogs. Okay. So there's also a lot of technical advantages about using frogs as a model organism as compared to, for example, mice or chickens or other things like that. So here's what our frogs look like. This is a female and this is a male. What's important to know is that they have what's called a diploid genome. So they have 2 copies of every gene, one from mom and one from dad. That's like humans. Other model organisms that people often use, like fish, have 4 copies. And so that makes really genetics a bit more tricky, and there's been a lot more shuffling of the genome that's gone on in those cases. So the species of frogs I use, in Tropicalis, has a diploid genome, and so that makes the genetics a lot more straightforward, and we're able to actually find, for example, the frog version of CHD2 very easily. What's really great about them is that they lay over 4,000 embryos a day. So each one of these little dots here is an embryo that's developing in the water. So we have a lot of embryos to work with. This is in contrast to, for example, mice, who usually have brood sizes of 5, maybe 10. So we're able to do a lot of experiments every day and move quickly. What's also nice is that now, with the advent of CRISPR-Cas9 genome editing, which you've probably heard about, we're able to go in and in a very targeted way, mutate any gene that we're interested in very specifically and cheaply. So in the past, when you would have to do really time and expensive experiments, we can now very cheaply and very quickly go in and mutate any gene that we're interested in. And then another unique advantage of working in frogs is that we're able to do high throughput drug screening. So for example, we can make that mutation that we're interested in the tadpoles, and then just add the drugs to the water, right? So these are aquatic animals. The tadpoles are developing in water. If you put a drug in the water, it will go into the brain. And so that's a real advantage here, is that we're able to do rapid drug screening to identify drugs that can reverse whatever we see in our mutants. So I'll give you one example of what we've done with that later on with a different gene, but it's certainly something we're looking at now in the context of CHD2. Okay, so this is our typical afternoon. Here are 2 frogs mating. Each one of these dots is an embryo, and here I'm showing you a time lapse showing all the first cell divisions of life for each one of these animals. And what's nice is that in the dish, we can watch every stage of development. We can watch the entire formation of the brain. And we don't have to, they're not in a uterus, for example, so we don't have to do any dissections to look at them. We can just look at them as they are. But maybe one of the cooler things about frogs, which is only true of frogs, this is a difference between other species, is that we can actually make half mutant tadpoles. So what we do is we take an embryo at the 2-cell stage, so it's only divided once, and the embryo has 2 cells. And so then into one of those 2 cells, we inject CRISPR reagents to be able to make a mutation in the gene that we're interested in. And we also inject a red dye so we can know which side of the embryo has been mutated. And then only 5 days later, what'll happen is you'll have a tadpole where only half of the brain has been manipulated. And then you can compare the brain. And so here are the eyes, here's the brain of the tadpole. I'll go into this in more detail in a minute. But now you can compare what has happened to this side of the brain that carries your mutation compared to the half of the brain that has not been manipulated, and start to understand what that gene does during development. And again, this is scalable since it is cost effective and quick. So again, this is what our afternoon looks like. We can make many different embryos where only one half of the animal carries a mutation of interest. Now this is a bit older stage where the brain is developing now. And again, you can find animals where just half of it has been mutated. And now you can compare what's going on in this half of the animal to the other. So it's well controlled, right? We're able to look directly at animals and say, okay, loss of the gene caused this to happen, because we know on the other side that didn't happen. Here's just one example to show you that we can do it. Here's a tadpole. These are its eyes. This is its nose. This is its mouth. Here's the brain. These are its ears. And what you'll see is on the right half of this tadpole, I've injected a CRISPR to mutate a gene that's required for eye pigmentation. And so what you'll see is on the side that I've mutated, you'll see an albino eye. It's lost the ability to make pigment. And you can compare that to the control side, or the side that hasn't been manipulated, where the eye is still pigmented. And so in this experiment, we say this gene is required for eye pigmentation. Here's what it looks like if we just image the brain, right? So what we want to do is we want to make these mutations, and then we want to image the brain and say what happened, right? And so here's one example of how we image the brain in the frog. Again, these are the eyes, the nose, the forebrain, midbrain, hindbrain, different brain structures. And again, what's cool about this is just another picture of the tadpole. I was showing you just the head a second ago. And so here in green, again, is that brain. There's the eyes. Here's the brain. There's the mouth, right? And what another advantage of the frogs is that we don't just have to study the brain. We can look at all different kinds of organs to understand what happens when we lose that gene. So the eyes, ears, again, kidneys, gut, muscles, heart. Since a lot of times these genes are not just doing something in the brain, oftentimes they're affecting many other organ systems as well. Okay, so let's get to the point. So one of the questions that came up in our work was, what does CHD2 do during embryonic development? And so this work is published. It was published last year in the journal Neuron. And so if anybody needs that paper, feel free to contact me. Happy to send it your way. And so we wanted to ask this question. And so one of the first questions you ask when you're studying a gene is, well, where and when is it on? So when is it active during development? And that gives you clues about what it's doing and what organ systems might be affected if you lose it, right? So the first question was simply, where is it during development? And so these are pictures of frog embryos over development. Again, those kind of first stages where the cells are just dividing, they haven't yet made a brain or things like that. And so everywhere where you see purple here is where CHD2 is on, where it's been made by the cells. And so here you see it in this ring. That's pretty typical for a gene that's going to be involved in brain development. Here, these are going to become the eyes, and you see this part right here and this part right here. Those are going to become the brain. So it's highly expressed in eyes and the brain. And this little chunk here is going to form really sort of the craniofacial skeleton of the frog. So you can see it involved there. Here you can see it expressed in the kidney and also the ears and the eyes and the brain, maybe also in the heart and gut. And again, but what you'll notice, what I'm trying to show you is that it's expressed in many different tissues. So this isn't specific to the brain. It looks like this gene is on and doing things in other organ systems as well, including the kidney and heart. So this didn't give us a lot of clues because it's pretty widely expressed. Okay, so the next thing we wanted to do then, since it was pretty broadly expressed, was say, well, what happens if we mutate it? And so to do that, again, I've been talking about CRISPR a lot, and we can talk about that if people have questions about how we do these experiments. But what we wanted to do is to be able to make a targeted mutation in the gene and then disrupt the gene and then look at the brain and see what happened. And so we're able to do that. We targeted our CRISPR to this exon, this part of the gene. And so if this is the beginning of the gene and this is the end of the gene, we targeted it right here for technical reasons. And these are our control sequences. And what you can tell here is that the sequence gets marbled, and that tells us that we made a mutation where we thought we did. With high efficiency. So every time we did it, we were able to, so this is the efficiency of this animal. On average, our efficiency was more like 80%. Anyways, what you all probably care about is that where this mutation is in the protein is here. And so many of you probably know CHD2 has many different functional domains within its protein. So different chunks of the protein do different things in the cell. These are probably people know about chromodomains. So these are the first two chromodomains. So this mutation is pretty early on. So we think that this is going to be a loss of function mutation, and it's at high efficiency. So this is a pretty severe loss of CHD2 when we do these experiments. And so the next thing we wanted to do was to use this tool and combine it with what I told you about earlier, where we can make half mutant tadpoles and then say, we inject this CRISPR that we know mutates CHD2 into just half of the animal and then image the brain of this animal and say what happens. So here's what it looks like. Let me orient you just a little bit. So this left half is the side that was uninjected, so that's our control. The right half is the half that was injected with the CRISPR, and that's now been mutated, right? And so it's pretty subtle, and you'll see it a little bit more dramatically in the next image I show you. But if you look at this region here, which is the forebrain, if you measure the size of this forebrain compared to the other side, this side is smaller. And so we wanted to know why it was, but okay, so let's step back and notice a few other things. The brain is pretty well patterned, right? So you still have a forebrain, midbrain, hindbrain. It's not like garbled or things are losing the right fate, right? You still have a brain. It's just that now this part of the brain has been reduced in size. You see a little better in this picture here. So now we're doing the same kind of experiment where CHD2 has been mutated on just half of the animal, but now you can see it a little better here, right? When we zoom in on this part here, which is the forebrain, you can see a reduction in the size here. And now I have two colors because I'm looking at two different kinds of cells in the brain, okay? So these green cells are the stem cells, the cells that are actively undergoing cell division to build the brain. And the cells in magenta here are what's called differentiated neurons or mature neurons, and they're no longer dividing. They're now actually starting to talk to each other and form connections. And what you'll see here is actually a reduction in the number of the neural stem cells and also a reduction in the number of these mature neurons as well. It's a little technical, but when you actually look at the ratios of these stem cells to differentiated neurons, what actually happens is that ratio increases. So the overall brain gets smaller, but in terms of the proportion of cells that are stem cells, that actually increases, despite the fact that the brain ends up smaller. So that was interesting to us. We wanted to, now that we found this sort of effect on this neural stem cell population, was to go then into human stem cells, neural stem cells in a dish, and now inhibit CHD2 with a technique called CRISPR-I, same kind of idea of inhibiting the gene's function, and say, do we see the same kind of effect where there's too many of these neural stem cells there? And so what we did see was that an increase in the number of these stem cells in human neural progenitor cells or neural stem cells compared to its control. So that did increase. Okay, so what I've showed you so far was experiments we've done where we've made these half and half mutants, right? And that's really nice to be able to pick up things that are maybe a little bit more subtle, like this decrease in brain size and this increase in the proportion of neural stem cells. But now what we want to do is also be able to look at a whole frog where all of the cells have been mutated. And so to do that, we've now made a line of animals, a family of animals, where they all have the same mutation. And so to cut to the chase, that mutation is in a similar location, but a little bit earlier, now in the second chromodomain, and it's a 20 base pair deletion. And what's especially nice about when you have a line now is that we can look at animals where the gene has not been disrupted, animals where they only carry one copy that's been disrupted and then one copy that hasn't, and animals where both copies have been disrupted. So now we can start to look at something that's probably more faithfully recapitulating what patients usually have, which is one functional copy and one copy where the gene has been disrupted in some way. And so now we're able to look at these three populations and ask, what has happened during development. We're starting with the brain, but we're starting to expand this work to other organ systems as well. And so what do these animals look like? So we're just starting this work. What we have done is take these animals and do what's called RNA sequencing. And so we take the brain of each animal, so ones with that have two functional copies, one that only has one functional copy, and then animals that have no functional copies, and sequence all of the RNA from the brain. And that's, we do this as kind of a first pass experiment to say, what's happened? What's different? What genes have now been expressed differently now that we've disrupted this gene? And so what was interesting is when we compare, for example, the RNAs that come out in the case where you have two functional copies versus one functional copies, we actually don't see any differences, and that's probably a power issue. We can go into the technical part of that. But when we look at the animals that have 2 functional copies versus no functional copies, now we can find quite a few differences. That's here, everything in red. But what's interesting is if we go 0 versus 1, we can find genes that are different. So the ones that I'm really focused on here for the purposes right now is thinking about genes that change between 2 and none and 1, which are these 9. It's getting pretty technical, but what we found here was that when we look at the genes that change in more than one condition and we say, what do these genes do? You know, what processes are they involved in? What we really start to see is cell division. So spindle assembly, that's actually the cellular machinery during cell division that splits the chromosomes, right? Microtubule depolymerization. Microtubules are that structure of the spindle that during cell division splits up the chromosomes, right? Again, microtubules, mitotic spindle, mitotic cytokinesis. These are all the processes of cell division, cell cycle arrest, right? So by and large, what we've seen so far is this really striking pattern that is really cell division that is showing changes when this gene is disrupted. And so what we then wanted to do is take this information that we gotten in frogs and again, go back into human stem cells, neural stem cells in a dish and say, can we see echoes of this in human stem cells, right? And so we saw all these terms about the spindle, right? During mitosis, the spindle is this, structure that splits the chromosomes, right? And so can we say, what happens to the spindle when we inhibit CHD2? Okay, and so on this panel here, these are our controls, right? So it's a non-targeting CRISPR. It means it's not targeting any gene, it's our control. Nothing's been manipulated. And what we're doing here now is looking for cells like this, where they're in the middle of a cell cycle, their chromosomes are all lined up, they're undergoing cell division, and this purple thing here, which labels beta tubulin, that's the spindle. So that's this microtubule-based structure that's actually splitting the chromosomes during cell division, where we saw that signal in our RNA sequencing. And usually what this structure looks like is it has 2 poles, one on each daughter cell, and then it has these long radiating projections that are actually interacting with the chromosomes that then will pull the chromosomes that have been duplicated into each daughter cell, right? So 2 poles and this nice structure. Again, this is a control. 2 poles, this nice structure. 2 poles right here, right? But when we started looking at cells where we had inhibited, and these are human cells, where we had inhibited CHD2, we saw a much greater proportion of cells where instead of having 2 poles, we now had 4, and the spindle was sort of abnormally oriented, or this, kind of third pole in the middle that definitely isn't supposed to be there, or these multipolar cells here that are like that. And so we're seeing echoes of what we saw in the RNA-seq data when we now go into human cells. I'm seeing these disruptions in cell division. I mean, so this fits very well with what we saw earlier, where we're seeing a smaller brain, but we're seeing too many stem cells. And that doesn't make a lot of sense, right? If you have too many stem cells and they're all dividing, you should have to see a bigger brain, right? But actually what we think is happening is that these stem cells are trying to divide to make new neurons, but they're getting stuck. Because they're unable to faithfully execute cell division, they're actually undergoing apoptosis or cell death. And so these cells are actually dying. And so then that ends up leading to a smaller brain. So that's very much where we are in this project. What I want to tell you about now is where we're going. It'll be quick, and then we can just spend the rest of the time on questions. So where we're going with this now is to start doing drug screening. So in the frog, this is a really nice process because, again, we have so many embryos from a single mating. And now that we have this mutant line, you could imagine that we could do a mating, have 4,000 embryos, and now apply many different drugs on these animals, look at their brain, image them, and then say, do any of these drugs actually change what happens to brain development in a way that restores what was lost when CHD2 was disrupted. And so we've done this in that paper I talked about earlier. We did it for a different gene, which was called DYRK1A. It's a kinase. But again, we saw the same kind of things in frogs that we're seeing with CHD2. With DYRK1A, we saw a smaller brain. We saw too many neural progenitor cells compared to differentiated neurons, and also these same sort of spindle defects with DYRK1A. So when we did this kind of work with DYRK1A and did a drug screen of 133 FDA approved compounds, we actually identified estrogen as a compound that was able to rescue that defect in neural stem cell development. As well as many other compounds, which may actually end up being relevant to CHD2 because they were compounds that affect spindle formation. And so these are very much things that we're working on now, not just in the context of DYRK1A, but in the context of other genes, including CHD2 as well. And we're now planning this drug screen to do on our CHD2 mutants to try to find FDA approved compounds that can reverse what we're seeing when CHD2 is mutant. The other thing we're starting to do now with CHD2, which we've done with that gene DYRK1A, is start to look at behavior of these frogs. And so in patients with DYRK1A mutations, they often see severe circadian rhythm disruptions, sleep problems. And so what we're seeing here is animals that are either control or that we've inhibited that gene DYRK1A, and we look at their activity over time. And this is about when the brain is done developing. Oh no, birth here. Here's about what we've mapped birth in a frog to humans. And about then is where we really start to see these circadian rhythm disruptions, right? Each one of these cycles, right? Awake, sleep, awake, sleep, awake, sleep. We start to see that they're hyperactive when DYRK1A has been inhibited. And again, now this is something we can drug screen against to see if we can restore these circadian rhythms, right? So we were able to see that estrogen rescued the stem cell difference in DYRK1A. Now we're asking, can estrogen rescue the circadian rhythm difference as well as gut and heart problems in DYRK1A? And so this is the kind of experiment we're also now starting, now that I have my own lab, to study for CHD2 as well. So hopefully I can come back in a year or two and tell you what we've found there. Right, so other organs, right? And so I told you a little bit about the brain, and now I've told you a little bit about what we're going to do with behavior. And so, one of the things I found with DYRK1A is that I wasn't really aware that patients with DYRK1A disruptions often have really severe gastrointestinal issues. And I didn't know that until I gave one of these kinds of seminars to the DYRK1A syndrome and the families that were affected by DYRK1A. And the thing that they kept telling me about was the gastrointestinal issues are very disruptive to quality of life. And so, I'd be very interested to hear if those are kind of similarly echoed for CHD2, since I do think that they're doing very similar things in cells. And we did see CHD2 being expressed in kidneys and gut and heart as well. So those are things that I'm looking forward to thinking about for CHD2 as well, now that I've kind of had that experience with DYRK1A. Okay, so let me give you a summary, and then we can spend the rest of the time on questions and talking. So the things I really want you to take away from today are that the human and frog sequences, but at the protein level, are very similar. So what we learn about the kind of core function of CHD2 in cells and in brain development can be directly applied to what we know about the function of CHD2 in human. CHD2 is on, it's active, and or it's been made in many developing organs, not just the brain. And so we're starting to think about other organ systems as well. And then what I showed you in the context of the brain is that we think that CHD2 is important for neural stem cell development. And without it, there are problems in cell division and specifically mitotic spindle formation that are probably leading to cell death and then reduction in overall brain size. And what we're excited to do is now use drug screening to be able to identify compounds that can reverse or rescue that issue in cell division. Okay, a lot of people worked on this project and helped. These are the core people, our collaborators, our funding, mostly from the NIH and the Chan Zuckerberg Biohub, as well as some private philanthropy. This is our website that talks a lot about the frogs. Here is my email. I encourage anybody, you're more than welcome to reach out to me anytime. I really value these relationships, and I learn more than any of you do. So please don't hesitate to reach out. More than happy to talk. So thank you again for having me, and I'm happy to answer any questions.
Thank you, Helen. We have one question in the chat. And the question is, how is frog brain development paralleled to human pre and postnatal development?
Oh, great question. So this is something that we worked on pretty extensively before we started this work, was to make sure that kind of the timing of events and the overall events were pretty well paralleled before we were going to invest all this time and energy in making those mutants. And so what we did is that we did RNA sequencing on the frog brain all through development. And then what we did is we tried to line that up with RNA sequencing that's been done in human brain development and say, do they line up in the same way? Are there any gaps that we're seeing? You know, when is birth in the frog compared to the human? And what we saw was that they lined up really well. And so we were able to actually say, you know, like when we were studying autism genes and we're saying, when we see all these autism genes being co-expressed, that really lines up to second trimester of human brain development. So we're actually able to kind of map in that way. Obviously, the human brain is a lot more complicated, and there's a massive expansion of the forebrain region in humans compared to frogs. But on the whole, if we think about just overall programs that are going on, so cell division comes first, and then migration, though there are exceptions, then usually migration, and then astrocytes are born, and then synapses are made, you know, those sorts of chronologies, and then things are myelinated, are paralleled and go in order in frogs as they do in human. That's a great question. Of course, there's differences, but the overall picture is very nicely paralleled.
Great, thank you. Go ahead, Christine.
Dr. Willsey, I had a couple of questions for you as I was following through your comments. So I don't want to monopolize, but I've just got two questions up front. You mentioned that different parts of the CHD2 gene have different functions. And so if, say, our child has a mutation or a variant in a different part of the gene, do you think then that, is it your theory that a patient's phenotypes are variant determined, or that they're connected?
Yeah, that's a fantastic question. So that's something we're really working on now, because a lot of times there are, you know, variants of unknown significance, or where it's not really clear what the effect on the protein's function is going to be just by looking at the sequence. You know, so some of these domains within the protein are really well understood. So some of them, for example, if it's in the nuclear localization sequence, right? That's the sequence that sends the protein to the nucleus, right? And so if it can't get into the nucleus, it can't do one of its jobs interacting with DNA. And so a variant there is going to be different, I think, than a variant at the very end of the protein, where it can still go into the nucleus, it can still do whatever function it's doing in the nucleus, but maybe it doesn't quite bind quite as well as it should to the sequences or whatever, right? And so where the variant is along the protein sequence, I think will change how severely affected the function of that protein is, and then also where it is how conserved that domain is, how important that domain is to function. Absolutely. Now, there are exceptions to that, which are, frame shift mutations or nonsense mutations or large deletions. So these are often, and that's what we made in our frog model, is a 20 base pair deletion pretty early on in the gene. And so that's really thought to be a non-functional version of the gene. Usually if it's that early and it causes what's called a frame shift and an early stop codon, usually cells will get rid of that transcript before it even makes a protein. And so that's thought to be a really just non-functional version of the gene. So historically, those have been easier to interpret. But yes, I think depending on where it is within the gene, will absolutely affect sort of which functions it can still do and which ones it can't.
Fascinating. One other question. You mentioned that the cell division process itself is altered by a mutation in the CHD2 gene. Coming from a parent's perspective, my child is 19 years old. Does that mean that any therapeutics, in your opinion, any therapeutics for CHD2 are not going to be helpful to an older patient because we're past the cell division process in the body?
So I don't know. That's something we're definitely working on, so when we're doing these drug screens, we're doing them pretty early, right? And so what we want to do next when we find something that can restore function is do them later and say, can we still restore some of these functions, right? There are, it's controversial in the field, but a lot of people believe that there are still neural stem cells later on in the brain that are active, right? And so if that's true, then there may be some stem cells that are still there that could function. But the other thing is it doesn't have to necessarily be cell division that could help a person, right? So mitotic spindle formation and cell division is obviously what I've been talking about, but that really reflects a difference in that structure that are microtubules. Microtubules make up spindles, but microtubules do a lot of things in cells. So it's not just cell division. They also traffic neurotransmitters out to synapses and neurons, right? So maybe we can't fix the cell division part, but maybe we can help them actually traffic neurotransmitters, and that might have some helpful effect, right? So I'm very hopeful about what's possible with, trying to be reasonable about it as well, right? So I don't know. Even if there aren't neural stem cells, you may still be able to help the end result because of other functions that gene does in the brain or in the gut or in other organ systems as well. But it's certainly something we're thinking about, right? Because a lot of people aren't diagnosed till later, or and so when can we intervene, and will it still be helpful, and how can we test that in the lab and think about that? Absolutely.
Mm-hmm. Great. Marnie, and I got follow up too, but go ahead, Marnie.
I was just saying, kind of along what you were saying with that, the DYRK1A, were you able to find, you said you were able to do some of that, and then did that help some of the kids that were already diagnosed and older?
So we haven't taken that into patients at all. So with that work, we only finished it last year in frogs. We validated some of it in human stem cells. So in human stem cells, estrogen was also able to help. With something like estrogen, that's such a big hammer. It has obviously other effects in sexual development and things like that. So that's not something that I would necessarily pursue on its own as a potential therapeutic. But in that paper, what we were able to do is actually say, well, why is estrogen doing this? What function of estrogen is causing this? And so what we then figured out was that estrogen is inhibiting a pathway that's called hedgehog signaling. It was just discovered in flies, so it's called hedgehog. It's beside the point. But there are a lot of targeted drugs on hedgehog signaling, and hedgehog signaling is a big cancer pathway. So there's a lot of drugs there. So that's the kind of work we're doing to maybe make it a bit more specific, make it something not involved in sexual development, find something that can be a bit more targeted to what we're actually looking at, and then start to think about moving that forward. But that we don't have any clinical trials or anything like that planned. We have a lot more work to do on the basic science front. And one of the things we're doing now is adding the estrogen later and saying, can it still rescue, right? Very much towards Christine's question of sort of when can we intervene and does it still help? So that's kind of what we're thinking.
Well, can I ask just a follow-up question for this? And at full disclosure, I'm not a parent. I work in the biotech industry, but I'm really interested in what you're talking about today. So if estrogen is somehow involved, and maybe this is a question to parents of girls, do you see a difference during sexual maturation? Does something happen to the course of the, the clinical symptoms that you see? Because we might be able to learn from that. And is this the case for DYRK1A?
Yeah, so what I'll say about that is with DYRK1A, and I think with CHD2, at least what I've read is there's not a sex ratio difference, as many girls as boys are affected by it. But that speaks a lot more to the nature of the variants. Really, de novo mutations don't seem to be impacted by sex because they are such large effect variants. They don't seem to be modified in that way. And sort of the amount of estrogen we're adding that can rescue is large. So I don't, I think in a natural occurring population, the effect of estrogen and sex is much more seen on common variants. We can talk about that a little bit more offline. Like in autism, for example, if you look at the entire population with autism, there's a very strong sex bias, 4-to-1 male to female. And so that fits with what we're seeing, that maybe estrogen is protective, but I don't think it's protected for these large effect variants. I think it could be protective on the scale of common variants that are adding up with each one individually, individual risks, but on a population level ends up there. Now with DYRK1A, in talking to families, they overall, it seemed as many girls as boys were affected by it. People did report some changes during puberty for girls, but they weren't necessarily helpful. It's not like there was a regression of symptoms during puberty, and then they were just sort of like things kind of changed a bit for some of their daughters. But again, that's an N of like 3 people told me that. So
yeah, this is something that we would probably ask if we do a community survey. That was a thing that we can add to a questionnaire, so we can see if it's something that is relevant.
That would be great to know. If you're going to do a community questionnaire, I would love to be able to talk to you about it a little bit before you send it out. Like we're trying to do that now with DYRK1A, and we don't want to keep asking these families to fill out these things, right? That's intrusive and it's time intensive, and nobody's got time, and so it'd be great to talk to you about that so that we don't both want to do the same thing and then not.
Yeah,
so
Christine, this is something, and Lauren, this is something that you can ask the citizen, that citizens look at as well.
Yeah,
the
you bet.
registry that you're running.
Definitely.
Yeah, we'll connect Helen too, as well as we already have you on board on that piece. So that's a good point. Thank you. Looks like we have some more questions. Brian, do you want to ask your question?
I think Jean and Luis was first.
Oh, it's just first of all, say thank you for having this today, and sorry I'm late, so I'm coming in very late. I have a 12-year-old girl with CHD2 variant, and we live in Southern California, so if there's anything that we can do to help, we can come up. She hasn't started menstruating yet, so if there's anything that you need from us prior to that, let me know. Happy to help, because we're all in this together, and we really hugely appreciate everything you're doing. And you can reach me through, I don't know how we do that here, but somebody can tell you, I guess, or I can put it on the chat. Is that okay?
Yeah.
Thank you, Jean. Yeah, it's a really kind offer. Thank you.
You can always contact us too, Jean, through Coalition to Cure CHD2 to help make that connection as well.
Okay, just if you want to post that, then I'll just do it right now, so it's done.
Sure, I'll put
That'd be great.
the email address in.
Thank you all.
Thank you.
Brian?
Hi, Helen. Thank you so much for the presentation. I was just curious because there's a chaser ortholog to in the frog. If you've done any work on chaser either individually or in conjunction with CHD2.
Okay, so remind me, this is like a long coding RNA that's thought to increase the expression of CHD2?
Yes.
Yes. So I'll tell you what I know about that. So I think it is conserved between humans and frogs. I think that's actually kind of rare. I think usually it's not between other species, but in Xanobus tropicalis, it is conserved. I looked that up. I haven't done anything on chaser. It could be done. What's easier to do in frogs is just overexpress CHD2. So what we do commonly is mutate a gene and then try to rescue it with, the human version of the gene. So we've done that for DYRK1A. We mutated DYRK1A. We then overexpressed the human version, and that rescued, that restored brain size very nicely. But then what we also did was just overexpress it even more. And actually then we saw an increase in brain size and DYRK1As in the Down syndrome region. Anyways, so we can do those sorts of things pretty easily with CHD2, just increase the dosage. And so I think, some families have variants that are thought to be gain of functions, or they're duplicated, or they have a small CNV that duplicates CHD2. And so that would be very relevant to that. We haven't done it, but I think we will do it in the context of just doing these rescue experiments, showing that the human gene can stand in for the frog gene. And so we should keep in touch because we do have those constructs. We have the human CHD2, we have it tagged in such a way that we can see it. And so I'll keep that in mind when we're doing those experiments to overexpress it and see what happens. Yeah. Thanks for reminding me of that. Yeah.
Just, I was just kind of curious. I heard you mention neurotransmitters. Has there ever been a study of like neurotransmitters of humans and any of these kind of autism, epilepsy related that you're aware of?
Yeah, so I don't know, CHD2, people are really focusing on its role in the nucleus and its sort of DNA regulatory roles. I haven't been doing as much of that. I've kind of taken an agnostic approach to what is CHD2 doing. So much of the field has been focused on its role regulating other things at the DNA level. So I don't think there's been as much on CHD2 and neurotransmitters. Certainly for other genes, there's been a lot of work understanding, what they're doing in that context and in the context of epilepsy. Yeah, and I'm happy to send you some of that work as well. But things that disrupt microtubules, that traffic a lot of those things certainly affect neurotransmitter release and can cause seizures and things like that as well. I see a question from the Willsey family about seizures and epilepsy. So that certainly fits with that idea, right? If you can't traffic neurotransmitters right, if you can't release them appropriately, that'll certainly oftentimes end up causing seizures. And so I guess since I've started that, do I see epilepsy? We haven't looked. So we've mostly been making these half and half mutants. And so when we do that, we just image the brain after we don't image them live. But we're going to start doing that. And now with this frog line, we're going to do that. What I'll tell you is that, we have now these adult frogs that only have one functional copy of CHD2. They look really typical. They mate, they produce plenty of embryos. I haven't noticed anything that looks like any kind of seizure activity, but that's certainly something we're going to be looking for, but haven't done yet. So that's for the Willsey family. Brian, did I answer your question sufficiently?
You did. Thank you so much.
Okay, good.
I have a follow-up question to Jean and Lewis. So is there anything that we as parents can do to help advance your research?
Yes. What a kind and generous offer. I mean, I think these kinds of conversations are really helpful for me in terms of knowing like just appreciating kind of everything that's going on. The idea that you all are interested in, and what do you do when the kid's already 19? Can you intervene there, right? That's really helpful information for me to know, because like, of course, academically, I can be interested in CHD2, but like, if I'm actually trying to help people, that's the kind of information I need in order to say, oh, I need to prioritize those experiments that are looking at intervening later. Right. So like, if there's anything, that you all can tell me about, like what would be a really helpful thing, that helps me prioritize what to look for. Right. So like, what are the things that are most disruptive? What are the things that you're noticing? And, I think these Facebook groups are amazing, right? You can actually share your experiences and get together and understand sort of what's in common and what's really going on. So anything of that sort is very helpful for me in terms of prioritizing my experiments, because I can be an academic all day long, but if no one cares about it, then why am I doing it?
So I think it's a great point, Helen. And one of the big questions about translating work from animal models into humans is, and this is why I asked the question about brain development, you can test in an embryo all day long, or you can inject in a baby mouse that was just born, but that's not analogous to a human brain of a child that was just diagnosed at the age, even of 3 months, it's already not the same. So how can we learn about what happens in the older brain and the older organism? Is CHD2 still important later on? Is it, and to Brian's question, and obviously he's coming from the other side, what happens if I have a drug that I can upregulate the normal CHD2, but I do it, I give too much? What are the consequences? So there are a lot of questions that we can ask. So it's endless.
Yeah, I mean, that's the real crux of what's going on right now in terms of thinking about gene therapy or pharmaceuticals, right? I mean, like, replacing a gene sounds great, right? But then the question is, well, where, when, how much, what's your readout? How do you know it's working, right? And then you have to do it for each gene, right? Whereas if we were able to take some of these genes and sort of understand that they're doing the same things in cells, like, for example, I think CHD2 and DYRK1A are doing a lot of the same things in cells. And so then can we say, well, this has worked for DYRK1A, and it's not about gene therapy or getting that dosage right. It's about using an FDA approved drug that works in that context. Can we now try it in this other one? Because we think the cellular functions and the expression patterns are very similar. But I appreciate, thinking about later stages, right? Because, I've very much been looking at brain development because I think that will give me a window into cellular functions and things that are the primary driver rather than a secondary effect, right? Because of course you could have that first thing happen and then something happens to sort of like mitigate that or interfere with that, right? And if you treat that secondary thing, it might help a little bit, but it doesn't get the root cause of what's going on, right? But I appreciate that idea of thinking about what we can intervene on later and still be efficacious. I mean, I think that's where this mutant line will be really helpful, because we can look much later and try that. But it's well taken. Yeah.
I just wanted to highlight from the Coalition to Cure CHD2 perspective to this group that's on the call, and we hope to do more educating to our community as a whole. But we are working with RareX and with citizens to have surveys taken of families to find out what are the areas of concern for our families. And Citizens provides the service of collecting everybody's medical records and extracting that information to help ultimately maybe create a natural history study so we can show what the disease looks like over time. So we want to get our families up and registered on those platforms, but we'd love to work with you. And of course, we're working with Yale to answer those surveys as well, because we want to get you guys the information that you need. I will just say that the GI issue is a big issue in the CHD2 community. It most certainly is. And I've noticed, as Coalition to Cure, we are involved in a number of other organizations as well. And with rare epilepsies, we're seeing that that's that's a big issue across the board, is these GI issues. So, and I think it's important that you pointed out the loss of sleep rhythm and the hyperactivity that you were seeing with this other gene. I mean, it resonates so closely to our experience. And I kind of think, you know, is the loss of sleep, the constant getting up all the time, every night, and not having sound sleep leading to the seizures, or is the seizures waking her up? I mean, it's just like a vicious cycle. But I too have a 19-year-old daughter, and I will say from our experience on the estrogen piece, that most certainly when she went through puberty, and we believe that there has been changes significantly, her seizures have increased with puberty, and they are linked to that time of the month. So I'm convinced that it is hormonal for sure. Yeah. So love to educate as much as we can, and we would participate in all of those surveys. So we just want to help make those happen.
Great,
Marnie?
thank you for that perspective. Well, real quick, I mean, like learning about the GI, you know, I have our project on GI development and thinking about some of these autism genes. So hearing that that's something that's a shared experience, you know, you know, it's not hard for me to say, okay, we've been doing this for 1A, let's do that same thing now for CHD2, right? So that's really helpful for me to hear, because now I can add that gene in for the stuff we're doing on gut development. So thank you. And it's hearing the experience in puberty that's not necessarily that it got better, right? But there is something there. There's something going on with sort of that process of sexual maturity. So thank you for sharing. Yeah, Marnie, hi.
I didn't say, first time I didn't say thank you. I just want to thank you for being here. But I'll just ditto what Christine said. I have a 15-year-old daughter. So we kind of had a similar experience where the seizures came back with puberty. And we definitely see it with the cycles. But one thing I've always wanted to ask, and this probably is not answerable, but I want to ask it anyway, because I never get to talk to people like you, is have you, and this is for most genes, have you been able to figure out like what triggered the de novo mutation? Like, is it even possible to find that out? Like, what could it have been? Something environmental? Is something we did, you know, when we were pregnant or something like that? It's always been a question I've wanted to ask.
Yeah, absolutely. And so what I'll say is that de novo mutations happen all the time. Most people have one or two. The question is just, where are they? Right. So it's a natural process that happens in the formation of egg and sperm. Right. It's why we change as a species over time. Right. It's a critical part of evolution to create new variants. And so sometimes those variants can give an evolutionary advantage. Right. And, you know, sometimes they end up in a really critical gene like CHD2. And you know what that's like. And so what I'll say is that it's impossible, I would say, to know why one of them happens. Of course, there are things that can change the number that somebody has. But by and large, it's just a numbers game of where those one or two de novos end up is usually what's going on.
And just to echo what Helen said, we all have mistakes in us. So it's just a matter of where the mistakes take place. There are some genes that are more amenable to mistakes than others because of their structure. I don't think that's completely known why, but in general, we all have it. So it's nothing that you did, Marnie, or you didn't do, or that extra beer that you had, or it's nothing.
Oh my gosh, yeah, no.
I feel like that's every mother's first question is, what did I do?
Of course.
I think actually the average genome has 7,500 structural variants. The average person, 7,500.
Yes. So when I say 1 to 2, those will be those are usually classified as loss of function, likely damaging variants. So there'll be a lot of changes, and a lot of times they're inherited from your parents. So inherited mutations, there's a whole lot more in each person, right? But in terms of de novo damaging variants, you just get a couple and they're random. But in terms of, differences, genetic differences in egg and sperm, there's a lot, but it's nothing you can control.
I just want to echo one thing, or perhaps say it originally. The huge reason we're so grateful to you, or I am, is because we're constantly personally fighting to find drugs that work from the bottom up, as opposed to using these umbrella drugs to try to ameliorate some of the behaviors. We've now on our second psychiatrist who wants to prescribe Abilify, and I just don't feel good about that. We're on guanfacine, but Abilify scares the bejesus out of me. So the fact that, I look at you as basically fighting against the kind of, I mean, everybody's trying to help, but it really, I don't think we know what we're doing with these young developing bodies. And if you're introducing something with the potential of like causing tardive dyskinesia, as in the case of Abilify, that is irreversible, just to say thank you again for looking at this from the granular level outward instead of the other way around.
Thank you for saying that, Jean. I think, the reason I got into this is because I think we need a lot more foundational research. I think psychiatry as a whole needs a lot more foundational research. We've stumbled upon some drugs that work for some people, but there's been no systematic way. And I think, now that we know these genes, that's the way forward, right? That's how we figure out what to really do here, right? It's a CHD2 and understanding all of your perspectives and the things that are going on with your children are that other key piece to understanding what to intervene on. So thank you for sharing your experiences.
Can I ask you, Helen, because you said like the gene it's like the long line and you can mutate anywhere along the line those, but I will say, I mean, so I know all of our kids probably have a unique gene mutation. However, we do seem to, in general, have very similar characteristics, the type of epilepsies, the type of behavior issues. And so, when you said, I guess when you first said that CHD2 is in every part of the body, in the gut, the kidneys, and that made me like, oh great, good luck finding a cure. But then the fact that all of our symptoms seem to be relatively similar, not always, but in a lot of cases, does that give you kind of more reason to kind of push forward on this gene, to think that, or is there similarities there that even though they're all, the line is, variations could be many, many? I don't know if that was a really good, clear question, but does that make sense?
It's a great question. It's a great question. So a lot of times, there can be a variant at any different spot. But a lot of times what's going on is just that you're just losing function of that gene, right? And so you can do that in many different ways. But each one of those effectively are having the same outcome, right? That gene cannot function the way it does if it's not variated, right? And so, so that makes sense to me that you could have variants in very different places, but have a similar human experience, right? Now, do I feel that because it's on in so many different organs, do I feel less hopeful? No, I actually feel more hopeful because I can figure out what this gene does in the gut, in the cells of the gut, and that can actually teach me a lot about what that gene does that I can then apply also to the brain. Does that make sense? And a lot of times if you're intervening with a drug, drugs don't just affect the brain, right? A lot of times they'll affect many different organ systems. But if that gene is doing the same thing in many different organ systems, maybe that drug will actually be tolerated better because it'll change that in all the different organ systems, right? Whereas if it was only on in the brain, you might have this off target in the gut, this off target in the kidneys, because it's not actually on in those. So I don't think of it as that's going to be harder necessarily. I just think of it as something that could give me more clues to understanding what the gene's doing. And then once we understand what the gene's doing, we can think about how to intervene. Does that make sense?
Does anybody have any other questions for Helen?
Helen, I don't, but I just wanted to thank you for spending the time with us this afternoon. It's really awesome. Awesome to see yet another well, a new model to us, at least, or to me. So cool. Thank you.
Yeah. Thanks for coming.
I have one question. It might be entirely inappropriate, so just say I can't answer that. But I'm curious about your title with the Chan Zuckerberg BioHub, an investigator. What does that mean? I just love the fact that somebody working on CHD2 is also working with the Chan Zuckerberg group, because they are so dedicated to rare disease that, you know, makes us feel like we're not off the radar in some dark closet and nobody's ever heard of us before. So just curious what that role is that you play in that organization, because they are so supportive of rare disease research.
Sure. So that's recent. I started being funded by them in March. So they do an initiative every 5 years to support labs at UCSF, UC Berkeley, or Stanford that they think are doing really high risk, high reward, pioneering work. And so I applied for that as part of this frog model to study these genes, including CHD2. And other genes associated with autism, genes associated with schizophrenia, and also Tourette disorder. And so they funded me through that mechanism. So me being an investigator there means that that they're supporting my work financially, and then I'm also part of a cohort of other investigators that have been funded for those same reasons. And so we get together every 2 weeks to sort of encourage that pioneering spirit, you know, they're not doing what everyone else is doing, and sort of think about how to solve these problems in different ways. So it's been a fantastic opportunity to have financial support to do things that aren't traditional. So that's the nature of that relationship.
Congratulations. That's super exciting.
Thanks.
All right, well, we thank you for your time today, and we're just so grateful for you to work on our favorite gene. So thank you.
Absolutely. Thanks for taking the time today. It was great to see everybody and hear your experiences. Appreciate it.